Hello and welcome to this week's What Were They Thinking? Among the many news items we have for you this week are Pfizer's new COVID-19 drug that is being described as repackaged ivermectin by those who have no understanding. Influenza has now started spreading through a Michigan University campus with the return of people to a somewhat normal life. Antibody discoveries? An interesting mRNA vaccine that's not yet been tested, but could prove useful for humans dealing with Lyme disease. A fungus that copes with radiation and as such could be used for spacefaring. Children's microbes in their gut seem to have no relationship to autism spectrum disorders, or at least according to one study. The timestamps for these and other news items can be found in the description box below. Let's start with Pfizer's protease inhibitor. This is a drug which is meant to stop proteins from being broken down. This can be done either within a cell or secreted into the environment. In fact, this occurs regularly in human bodies. In the stomach and the small intestine, there are a number of proteases that are essential for us to be able to absorb amino acids from protein and then utilize them in our own muscles, bones, and cells. Proteases are essential for the human body and in fact for just about any other organism to be able to use protein and break it down into usable elements. Without those usable elements, notably amino acids, there is no way for the organism to be able to rebuild its own protein. These proteins are often essential for things like channels and gateways in cells. It's why protease inhibitors have been looked at as a strong way to deal with things like malaria. By stopping the protein channels that malaria uses to get into red blood cells, it is possible that it could stop working as a parasitic organism. The same theory, to a certain extent, underlies Pfizer's new drug called Paxlovid. This is supposedly nearly 90% effective in preventing hospitalizations due to COVID-19. The fundamental nutjob theory is that both ivermectin and this drug are protease inhibitors. Because of this, ivermectin, the not approved and not COVID-19 relevant protease inhibitor, is being suppressed. All of this is so Big Pharma can generate big profits. Yeah, this is kind of like saying that a monkey and a human are similar, but it's even more. It would be like saying that a dolphin and a human are similar. Yes, they're both mammals, but they both have very different organism structures and mechanisms within. For example, one of them lives in water while the other one can barely survive in it. In this particular scenario, Pfizer's new drug and ivermectin differ significantly. Pfizer's drug is based on ritonavir. This is a protease inhibitor used primarily for HIV and AIDS treatment. In this case, it targets some of the essential polyproteins that are needed to take viral DNA and turn it into protein that is useful to the virus. By inhibiting these proteases, you fundamentally stop the creation of new viral proteins. Without that, the virus cannot go on to do things like infect new cells using the spike proteins. Ivermectin does something vaguely similar, where Pfizer's drug targets specific viral proteins. Ivermectin is more broad, and it is more similar to malaria in that respect. It targets transport proteins. These are not necessarily specific to virus proteins. In that respect, yes, they are similar in that they do something vaguely similar, but in very different ways and with very different specificity. For this reason, although yes, Pfizer's new drug is a protease inhibitor, it is nowhere near the same sort of protease inhibitor as ivermectin is. Further to that, this drug can reach safe levels in blood plasma where it can have that effect. Ivermectin cannot. Ivermectin needs to reach levels in excess of 20-fold safe levels to be effective in plasma. That means it needs to be 20 times stronger to get around your body and attack the virus that has infected you. While still on the topic of viral infections, but this time a different coronavirus, influenza. Influenza has gone through the University of Michigan, creating 528 cases. This is only a year after there was less than 100 cases reported as serious infections across America. These were effectively cases that were so bad they were identified and sent to the CDC to be classified. That is a tiny, fractional number compared to previous years. All of this was a byproduct of the various measures taken: social distancing, mask wearing, better personal hygiene, and so on. 528 cases since October 6th, and that's a little over a month and a half. That's a huge number. Admittedly, this does come on the heels of expansion, or more accurately relaxation, of the various rules that have existed in the states in America over the last 18 months. Further to that, there's another significant factor. The majority of those who have been infected did not get the flu vaccine. In fact, 77%. This is why there are more infections than you would see in a normal year, despite this being the period where influenza would normally start showing up and expanding. This week sees the continuation of viral news. Pun intended. It's actually a case study of a woman who has cleared HIV from her body without any treatment. Understand, this is not an example to follow, but an amazing case study. As a general rule, HIV is incredibly hard to treat, primarily because it hides within the immune system itself, the very part of your body that is meant to deal with viral infections. As a result, even if the immune system clears out all of the other cells that are there that have been infected, it does not try to destroy itself. As a consequence, it will be knocked down and go into hiding for a while. Once the immune system rests, it will pop back up again and begin infecting cells, and so the cycle repeats. This is why HIV cannot be cured at present. The patient who was first diagnosed in 2013 has had 8 years of follow-up. They've also had 10 tests conducted. There is no evidence now that they have a viral infection, which is weird. Unlike the other cases that have cleared HIV from their system, this patient did not get a stem cell transplant, and as a result, there's no current explanation as to how they managed to clear the virus from their body. The challenge with this is proving just what has happened and then figuring out how. It's really not easy to be able to analyze all of the cells they need to analyze, get enough data from them to show that they have been completely cleared of HIV. Further to that, there's no definitive way of showing that they have been completely cleared, even if they find that with the cells they can get. The problem largely is the way HIV will hide, and the fact that the body can't generate antibodies to it that will keep on working. This takes us to our next piece of research, and that is antibody research in mice. Finding antibody research involving mice is kind of like finding arseholes. Everybody has one, so you're sure to at least identify one. In this case, it's the research that ties into creating a vaccine for Alzheimer's disease. Alzheimer's disease is one of the more serious leading ways to develop neurological or neuropathologies that wind up with your mind going. It's perhaps one of the cruelest fates you could have. Alzheimer's itself works by proteins building up in the brain and then blocking the neurons from transmitting signals and interrupting neurons themselves. a vaccine that could target that and the other mechanisms by which it develops could then be a leading contender compared to our current treatments, which at best can halt the progress of the disease and, in worst case scenarios, do nothing at all. There's also everything in between. The way this is going to work fundamentally is the same as for any other vaccine. It gives the immune system a small sample of what they're going to target, in this case, tau proteins. These are then going to get picked up by the immune system, and the immune system takes them off and develops antibodies as the initial way of flagging these structures as being something that needs to be dealt with. The site on these where the antibodies bind to is called an antigen, and depending on what you're looking at, there may be one or more antigens on the surface. This gives the opportunity to train the immune system to be very specific. Being able to vaccinate people before they develop the disease is great. What is even better with this research, although it is very early on and only based on animal studies, is that it can recover some degree of brain function, memory, and glucose metabolism, all of which are needed for brain function, the ability to be able to keep your brain doing what it needs to do and mitigate some of the symptoms and signs seen, and also, hopefully, keep the brain working well in the long term. Still focusing on vaccines, but moving away from viral to instead a different disease that is somewhat fringe, Lyme disease. This is using a combination of relatively modern technology that's well established, mRNA vaccines, and application to an existing disease. Lyme disease is generally picked up from saliva in tick bites. This then gets into the human host and it takes seat there. What's entered the human host is a small bacterium called Borrelia burgdorferi. This is a relatively easily dealt with bacterium. Antibiotics would deal with it, and as a result, you can easily get rid of it. The problem is repeated exposure, which could be a nuisance. This is where the idea of vaccinations come in. As with other diseases, such as, for example, smallpox, we're able to vaccinate people and preempt infection. And this is not just for viruses, but bacteria as well. The vaccine uses a small sequence of RNA, which creates protein. And in this case, the protein uses a very specific sequence, something which should be able to target the exact area needed to be able to deal with this particular bacterium. The way it does that is that it focuses on 19 proteins that are present in the fluid that comes from the tick when it bites. This is an area where there's already immune reaction in response to the tick bite, so they're really only accentuating it, but they're accentuating it in such a way that it allows the immune system to pick up on the Lyme disease causing bacterium and deal with that along with the tick bite. Unfortunately, this is also another case of very preliminary research. In fact, the evidence is only based on what's happened with guinea pigs and being bitten by ticks. That's really early in the research pipeline. As a result, the development of this vaccine is a long ways off, but it does propose an interesting way of dealing with these things. Rather than vaccinate directly for the pathogen which causes the disease, vaccinate for the delivery mechanism by which it's introduced into the body, and by virtue of that and developing an immunity to it, you create immunity to the consequential infection. Going now from diseases that are based on viruses and treatments involving vaccines to instead a somewhat different kind of disease, diabetes. Diabetes comes into two forms. Type 1 being where the body does not produce insulin, Type 2 where the body either doesn't produce insulin or more likely is not responsive to insulin. In the first instance, it was clear that something had to be happening in the body to prevent insulin from being made. In this case, beta cells within the pancreas are destroyed, often by the immune system. This has led to various theories that it could be possible to either introduce these same cells back into the pancreas, or it could be possible to stimulate the cells that are in the pancreas to turn into new beta cells. Both required a way to be able to get beta cells that could produce insulin. The current research looks at being able to use stem cells to create new beta cells which could either be introduced as stem cells or injecting stem cells directly. By using stem cells and then giving appropriate indicators on how they're meant to turn into the exact cell required, you can control the process. This is, however, very difficult, and that's where the research has been working. How do you ensure that you get the maximum number of beta cells rather than getting other cell types you neither need nor want? The way they're doing it in this research is to control the skeleton within the cell. This is called the actin cytoskeleton, and by controlling that, they can control, to a certain degree, just what the stem cells will turn into as they proliferate and differentiate, specifically trying to drive them towards becoming beta cells. The research has shown that although the process is difficult, the results are at least reliable. Reliable in so much as that they've lasted for at least a year so far. Type 1 diabetes, of course, being something that starts in humans very early in life. Thankfully, not a few cells early, but this new research could help examine just what's happening at that time in the development of the human embryo. You start as what is effectively half a cell, either a sperm or an ovum. The two come together and you have a full 46 chromosomes. From here, that one cell begins to multiply and turn into many different cells. These eventually specialize and become different parts of your body. Those parts of your bodies become tissues. Tissues become organs, and organs become systems. The process of going from half a cell to who you are now is not simple. That is why stem cells, such as those used in the diabetes research, can be very complicated and hard to manage without very, very strict controls. We're fortunate that we can examine much of what's going on in people who are alive. Embryonic research, not so much. There are many issues around the ethics involved in such research, and it's why, for the most part, they only ever really develop up to a 2-week limit. The research in this case is not derived from a laboratory-generated sample, rather an embryo that had to be aborted. Because of that, it was a rare opportunity to see just what is happening, and at this time there were approximately 1,000 cells. 1,000 cells that had already shown that they were beginning to differentiate and specialize. This embryo is only approximately 3 weeks old, but what is important for research is that it does match with what's been seen with mouse and ape embryos at similar time frames. That means that it's likely that these are extremely good models of what's happening in the human body, or more accurately, human developing embryo, as anything else we're likely to find. While many diseases aren't going to start at this stage in the development of the human embryo, they are going to start while the human is an embryo. The developing fetus is a very delicate structure. Small disruptions can cause lasting changes. For example, a small increase in testosterone during gestation leads to male offspring, whereas without that, female offspring. Other things can occur, such as neuropathologies, and it's in fact notable that things like autism can be identified in children while they are still in utero. There have been past examples of this that we have identified. This is important for, say, things like autism. Autism is often blamed on various justified and unjustified factors. Unjustified being things like vaccines, justified being things like genetics. One of the leading theories for quite a while now has been on top of this. The microbiome of the GI tract in autistic children is extremely important. This has led to theories that if you provide fecal transplants, children with autism may get better. The research that we're going to talk about in this part of the video looks at that, but believes that it's the other way around. Microbiome in the GI tract does not affect the brain, it's the pickiness about what is being eaten that changes the microbiome of the GI tract. So rather than being GI tract and bacteria affecting brain, it's brain affecting GI tract and then bacteria. The study is largely observational in terms of the way it went about this. It didn't try and set up an experiment where it could have different children in different streams. Rather, it worked with the Australian Autism Biobank and data from the Queensland Twin Adolescent Brain Project. It did involve a relatively large number of children for what it is, 99 children on the autism spectrum, 2 groups of non-autistic children, 51 siblings, and 97 unrelated children. This means that you had a relatively large and incredibly well-matched cohort, which was able to adjust for things like clinical diagnoses, family, lifestyle, diet, location, and other things that often become confounding factors when examining this sort of research. What they found was that there is no relationship between autism and the microbiome, or microbiome diversity. This kind of research is really important, especially when we refer back to that earlier mentioned fecal transplant therapy. If you're going to go and wipe out all the bacteria in someone's GI tract and then put new bacteria in there, you're going to propose a whole range of issues. Notably, the body's not used to dealing with those bacteria yet, and they may become pathogenic through opportunity rather than ill intent. Because of this, children who are picky eaters may be picky eaters because their body can't cope with various things, whether that is the sensory input or the consequences of eating food that they're not used to. Going from the microbiome of your GI tract to instead other microbiology related news, and in this case, the possible application of a fungus that would help with radiation during space travel. There have been many statements made about the possibility of returning to the moon, creation of a new space station that's used by various countries, and even going so far as to colonize Mars. All of these are very difficult things to achieve. The biggest problem is going to be just how do you get there without having to worry about radiation? Space radiation is everywhere, and it's a big problem. This is one of the reasons why astronauts on the ISS should only stay in orbit for up to a year. The exposure to radiation, while not as safe as it is on Earth, is higher, but not so much as if you were further out. The Earth still provides some protection. This is where a fungus kicks in, the Cladosporium sphaerospernum. What this does is use radiation through a process called radiosynthesis to grow and develop. It uses radiation, which is already energy, like plants use sunlight, to create what it needs to live. Of course, this is not a silver bullet of sorts to solve all radiation-related woes. The sheer thickness and density of the fungus requirement in space, or for example Mars, is prohibitively thick, and that is the only way you would be able to use it as the sole solution to radiation. This means it's going to have to be one of a number of solutions used in tandem. The final piece of news we have for you this week is still somewhat space related, but that There will be a lunar eclipse over the coming night or thereabouts, depending on when this video goes live. For most people, it'll probably be late Thursday night into early Friday morning. The lunar eclipse will not be a full eclipse, which is unfortunate. It will be a partial eclipse, but then again, partial in this case is something of a misnomer as well. It'll be 97% coverage, and frankly, most people won't be able to identify the difference. The lunar eclipse is going to be quite long in the making, and this owes to its location. Much like we had with the supermoon eclipse a few years back, this one is occurring at the moon's apogee. The apogee is where it is furthest from the Earth. Combine that with how the shadow is generated on the moon, and you can imagine it will last for a much longer time frame. It has to. The Earth is in between the Sun and the Moon for a much longer period insofar as the direction of light travel goes. What is even more interesting is that generally lunar eclipses and solar eclipses will correspond to each other. Often they'll be a small fractional time frame apart, again relative terminology, in this case a few weeks. It's expected sometime around early December there should be a solar eclipse. Unfortunately, this will only be visible from the southern hemisphere. Or does that even exist? What did the flat earthers say? That's all the news we have for you this week. Thank you for watching. If you found it interesting, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions that you have below.